Hello everyone, I am Miroslav Kratochvil. I'm from Luxembourg Center for Systems Biomedicine, and in this short overview I'm going to talk about COBREXA, which is one of the core software packages of the European project for the Personalized Medicine Center of Excellence. The main aim of COBREXA is to enable the metabolic modeling on HPC in the scale that is required to support various new and hypothetical future use cases in the personalized medicine. Let's first have a look at the brief timeline of the project so far. The COBREXA acronym stands for Constraint-Based Modeling and Exascale Analysis, which is an exascale play on the previously existing group of methods that are usually called COBRA. The main aim of the methodology is to represent the complex metabolic networks in the organisms as constrained linear systems and gather insight into the properties of the organisms by applying the associated linear methods, mainly through various methods of linear or quadratic optimization, and usually with high-dimensional sampling of the feasible space. We have developed the package from scratch at LCSB with much help from our collaborators from Heinrich Heine University of Düsseldorf. The package is relatively fresh. The main development has started around the beginning of this year, and the first public release actually happened in April. Despite of that, the package is already useful for many purposes. Mainly, you can now run your favorite COBRA methods on HPC without much effort. We have aimed to produce a kind of batteries co-included solution for parallelization of whatever workloads that may be parallelized. COBREXA is now available from GitHub, also from the official Julia package repositories. You may visit the website and browse the comprehensive documentation to see a complete list of the available features. Let's have a quick look on what you can actually do with COBREXA now. Traditional constraint-based modeling problem is to ask questions like how much of some metabolite is my small organism going to produce in some certain conditions, or perhaps what are the best conditions for the organism to produce as much metabolite, as much, for example, biomass as possible. This problem can be complicated with added variables. For example, you may consider various genetic modifications of your organism or additional chemicals and drugs in the environment. You can also combine multiple organisms of different genetic equipments and generally of different species in one community environment, or add an environment that has metabolism of its own, such as soil microbiome or human organs. Now, if you continue adding the complexity, you very quickly hit a problem size that reaches the exascale computation volumes, at which point you don't have many other choices than using the HPC resources, and that can be very easily done with COBREXA. The main role of COBREXA in personalized medicine is to run this kind of large-scale analysis on models constructed from human organs and basically any health-related models. There are publicly available models of human metabolism and related metabolomes that can be personalized to match the precise phenotype that occurs in a patient using mainly the genetic sequencing methods. With that, you can simulate many metabolic processes in the patient that gives you an opportunity to actually predict various events or treatment outcomes, such as various interactions of drugs with organs and microbiome. Or in more advanced case, you might try to backfit a partially personalized model to measurements from a patient to gain a more precise idea about what can be the underlying cause of what you observe. Within the project, our main task is to improve the usefulness of the metabolic models. One way for doing that, which we are currently working on, is the integration of COBREXA and COBRA methodology with the other core applications from PerMedCoE project. Mainly, we aim to connect COBREXA to a spatial cell simulator called PhysiCell to get more precise information about the possible metabolic outputs of the cells at various positions in the organism, which is especially interesting, for example, in solid tumors where the environment will become very unfriendly and deficient of nutrients. In the future, we aim to connect other sources of information that would improve, for example, the prediction of cell signaling outcomes. And that is everything from me today. Thank you for your attention. I'll be looking forward to the discussion at the PerMedCoE conference booth. Finally, I would like to thank all the contributors and colleagues from all the consortia and institutes that have contributed to COBREXA development, especially to my colleagues from LCSB Luxembourg and to our collaborators from Heinrich Heine University from Düsseldorf.